Okay, so when you take a look at cutaneous innervation, we talk about receptive fields. So a receptive field is the area of skin that's innervated by one single receptor cell. So we know that receptive fields tend to be smaller distally and larger proximally. So, like in the back, those receptive fields are bigger and there's less of them. And when we talk compared to like receptive fields in the fingertips, those are more distal joints. So we see those receptor fields tend to be smaller. And the distal regions of the body also have greater density. So they have more receptors in the distal areas than in the proximal areas. So distally, the smaller fields and the fact that there are more receptors there result in a greater ability to distinguish between two closely applied stimuli, like on the fingertips versus the back. So let's take a look at the next picture. So here we can see the receptive fields, right? So again, if the receptor field, let's look at receptor field number 1. So that's the area of skin innervated by nerve number 1. Right next to receptive field number 2. Now, so let's say that's the fingertip. So on the back, receptive field number 1 would be the whole space, would be a much bigger space. So why? Again, this comes back to the sensitivity of the fingertips, right? We use those fingertips and our distal joints for really specific fine motor control movements, those fractionated movements, right? If I need my thumb and my finger to button a button or pick up a coin, I need them to be very, very sensitive. What else did I want to tell you here? So each, hold on a second. So another example of why we need more sensitivity and smaller receptive fields and a higher density of those receptor fields in the fingertips, or like for reading Braille, right? When you're reading Braille, you are trying to discern these little raised dots on paper for reading. So the fingertips need to have finer 2-point discrimination than, let's say, on the back. So when you take a look at this picture on the right, they're looking at 2-point discrimination. So the little tool A on the left here over the blue circle, since one receptor cell is responsible for reading all of the contact in that blue circle, and you have a tool that's making contact within that blue circle at 2 different places, because only one sensory receptor is reading that whole area, the body and the brain reads that as one. It can't discern that those are 2 separate points of contact. So the other word for that is it can't discriminate between those 2 points on that tool number A that's sitting on the blue dot. But when you look at B, now B has 2 dots, right? And it happens to be placed over 2 different receptor fields. So that means 2 different receptor cells are reading each point. So your brain would recognize B as 2 different points. It would discern or be able to discern or discriminate the fact that those are 2 distinct points of contact with your skin. So this is an example. OTs do a lot of 2-point discrimination tests, and I'll show you the tool at the end of the lecture, because they are really interested in how sensitive, what the sensation is in your skin for all of the specific OT activities they're going to be working on, things like buttoning a shirt, picking up coins, all those things that you need to do, those fine motor skills. So arriving sensory information. So when we have sensory information, incoming stimulus can take many forms. Sometimes it comes in because of physical force, such as pressure, like when I put my thumb on your skin. Sometimes the information is coming in as a dissolved chemical, right? If you have, oxygen or pH, if that chemoreceptor is reading that information. It can also come in as sound or light. So regardless of the nature of the stimulus, the sensory information that's sent to the central nervous system comes in the form of action potentials, right? Regardless of the nature of the stimulus, that incoming information is all coming in from neurons that are all firing action potentials. So when we talk about transduction, that's the translation of the arriving stimulus into an action potential by the sensory receptor. So transduction begins when a stimulus changes the membrane potential of the receptor cell, right? So you could go back to our early discussions on generation of action potentials. So we have this receptor potential, which is the name of the membrane potential change that's caused by the stimulus. So depending on the stimulus that's coming in, it can either be graded depolarization or graded hyperpolarization. Again, the stronger the stimulus, the larger the receptor potential. The generator potential is the depolarizing receptor potential. So again, we've got dendrites of sensory neurons. Those are typically the receptors, right? The dendrites for general senses. And the sensory neuron itself is the receptor cell. Some of that may have been obvious. I just wanted to make that clear. So we also have those somatosensory projection pathways, which we talked about earlier in the semester, which we're going to talk about again. So from that receptor cell, that neuron, that sensory neuron, to the final destination in the brain, most somatosensory signals have to travel by way of 3 neurons. So we call those first, second, and third order neurons in this pathway, in these pathways. So the first order neuron brings the information from the sensory receptors into the spinal cord. The second order neuron brings the information from the spinal cord or the brainstem to the thalamus. And the third order neuron brings information from the thalamus to the cerebral cortex. And of course, we have a couple different somatosensory pathways, but that's kind of the general rule. That's kind of the general 3-step process for these somatosensory projection pathways, which we're going to talk about in a little more detail. And again, as we know, most of these sensory signals cross, right, to the opposite side of the hemisphere, to the opposite side of the body. Now, they cross at different locations depending on the information that they're carrying. Some cross at the spinal cord level, many cross at the medulla. But that's why, again, we have that contralateral symptoms from, wherever, depending on where the pathway damage is or the damage in the central nervous system. Oftentimes, the symptoms that we see are on the opposite side.